In this video, we will discuss about the AND gate efficacy on different biomarkers. In the previous video, we have understood that their AND gate system work on the initial biomarkers that they have selected, that is EGFR and PDL1. However, they want to ensure that their AND gate system works on different combinations of the biomarkers, and it can provide efficient and specific signals to trigger the downstream signalings in the tumor cells. So the scope of this video is to check the AND gate efficacies on different combinations of the biomarkers. So compared to the biomarkers that they have used previously, they have changed one of the biomarkers, that is PDL1 to PTK7, because aptamers that can specifically target these PTK7s are available. So they checked EGFR and PTK1, whether when both of them are present, whether they are able to induce the signaling, and the signaling that they have checked using fluorescein reactions or using fluorescein readout. So here also the initial testing cell line is A431 cell lines. So when they checked it in this A431 cell lines, they are able to see fluorescent signals when both the biomarkers are present. When only one biomarker is present, they are not able to see fluorescent signals. And when both of them are not present, then also they do not see fluorescent signals. So this indicates that the AND gate efficacy system works efficiently even with different biomarkers. But however, they also check their system on different cell lines, that is MDA-MB-468 cell lines. So here the expression levels of the PTK7 is low, so the fluorescent signals is low. Whereas for HCT-116 and MCF-7, none of the biomarkers are present, so they do not see any of the signals. So their system works when both the biomarkers are present. Even if one of them are present in the low amount, they are able to see its intensity decrease, fluorescent signals intensity decrease proportionally. And if none of the biomarkers are present, then there is no signals that they are able to see. And these things they have quantified in the panel E, where they can see that when both biomarkers are present, then only the signal intensities are high, and which is statistically significant compared to the control conditions when only one of the biomarkers are present or none of the biomarkers are present. Once they validate their AND gate efficacies on other combination of biomarkers, now they also checked whether the AND gate system work on single biomarkers or not. So in that case, what they did is they split the DNA initiator between the same Affibody or Aptamer of the same biomarkers. So here they check the system in the same cell lines, A431 MDA-MB cell lines, and this for both the biomarkers, that is EGFR and PTK7. For EGFR, the results shown in the panel C, where we can see that when both the biomarkers are present, then only they are able to see signals for both A431 and MDA-MB-468 cell lines. When only single biomarkers are present, or like for control they used different biomarkers also. So when only single biomarkers are present, they do not see signals. When it is combined with different biomarkers, they do not see any signals. And similar results they have observed for PTK7 also. Since PTK7 is expressed in high amount in the A431 cell lines, they are able to see signals when both the biomarkers are present. But for MDA-MB-468, it is present in low amount, so they see less fluorescent signals. And similar results they are able to see that in the case when only one biomarkers are present, none of the signals are no signals are expressed for the fluorescence. Almost negligible signals are expressed for fluorescence. Sorry. And in this case also they also use the control, where they use the biomarkers for the other where they use the different biomarkers along with this PTK7. So in that case also they do not see any signals. And these things that in panel C and D are quantified in panel F and G respectively, where we can see that intensities are higher when both the biomarkers, conjugate biomarkers, are present for both A431 and MDA-MB-468 cell lines. And similar results they have seen for this PTK7 biomarkers for A431 cell lines and MDA-MB-468 cell lines. So this indicates that the AND gate efficacy works on different combination of biomarkers, and it can also be used for single biomarkers detection with more efficacies. That is, when the single biomarkers are present more evidently on the cancer cells. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.